Okay, thank you very much for staying, folks. I will start this with a small interactive session, and if you do well, there will be a lunch exit option 2 minutes into my talk, okay? What is this man doing in this field here in the middle of Ireland? What's he doing? He's sampling earthworms, exactly. Earthworms are rule, okay? If you've missed that, who here also has sampled earthworms? Hands up. Excellent. Lots of people, okay? Lots of people. If you have seen it in the mega sessions of the posters, okay, folks, even if you are into minor sort of organisms, there is lots of research all over the world, and the issue really is why not amalgamate them, okay, and bring them all together. Let's write a review, okay? They're highly cited, so you will have impact, which is excellent. And basically what you can do is a narrative, okay, which is the old school, so you read stuff and you meet people and you're an expert with your own opinion, or you count articles on the same investigation, and you say, okay, for example, in this one we had 8 studies and 6 were this way and 2 were that way, which means it must be this way. Or you do it in a statistical sound way, which is known as meta-analysis, and basically the difference is that you weight the individual studies according to how large they were and how variable, okay? Because not each study is the same. Okay? So here, what you do basically with this tool, okay, you are not, within a narrative, but you actually extract data on treatments which have, from different studies that have investigated the same thing, and you build a new metadata set which you then analyze with statistics. You did very well in your test, okay? So this is my take-home message, okay? That this is a tool you could use in your own area, and indeed we have seen many examples in the last few days. Nobody leaving, that's very kind, okay? So if you are interested in the next few minutes, I will talk about one example, okay, where we have looked at tillage impacts and earthworms. So it is an obvious choice because tillage is a major land use factor, which we are very interested in. Earthworms, as we have seen, are particularly nice because you actually see them if you have to interact, with farmers and so on. You can show them, and there actually is an enormous number of articles, including, by soil scientists and engineers and agronomists who always seem to count earthworms along with what else they do. Okay. Here is just one example of my own work where we looked at different systems in England in large replicated plots, and we looked at, you always need one control, which is your standard farming system or conventional. Then we also had direct seeding, and you can see the residue on the surface. And in this example, we also had an understory treatment of a legume. Okay, and if you look, the results here are what you would expect, that you have a small increase if you reduce disturbance, and you do have a large increase when you also feed them, okay? But the thing we have to ask here is, so this is only one study, but is this generalizable, and what about other soils and so on? We have, of course, many excellent narrative, expert chapters and books and so on by, for example, Ken Lee and Clive Edwards and so on. But surprisingly, none of them have used meta-analysis. And this is why we started this. And as I said, you can formulate testable hypothesis in this, which we did. Okay, so we said if you reduce tillage, if you look all over the world in all the studies with all the different soil types and so on, we also expected that there will be impacts, of course, of the soil texture, rainfall, length of the experiment, and so on. Okay, so the method bits are the hard work that you have to search in databases and in the list of books and so on to find published studies which match your topic and which will also meet your expectations in terms of field design, and replication, and reporting of an estimate of variation which is often missing. And you extract the data and you build this metadata set. Okay, the statistic itself that is normally used is known as the response ratio, which is the ratio of your mean of the treatment which you're interested in over the control And this is nice because it is intuitive and it is reportable as a percentage increase. But unfortunately, we had a number of studies where we had zeros in the control, which means that we had to use HSD. And if you look here, basically what that is, it is the difference between your treatment mean and your control over the standard deviation. Okay, so in the results now you will see the effect size as E. Okay. Okay, so here you can see when you look at the abundance of earthworms that we had about 120 sources, mostly journal articles and proceedings. Many of them have included more than one treatment. So let's say they would have minimum tillage and also reduced tillage and so on. So in that instance, we ended up with 251 data rows, which was nice. And you can see it was a reasonable international spread with, if you can see this here, New Zealand, Australia, North and South America, Europe in the middle, and a small number of Asian and also African studies. Okay, here's a typical result. Okay, so you would always look at your overall mean, and the error bars are the confidence interval, and if they don't overlap with 0, this is a significant effect. And the first result you see here, if you look at the texture of the soil, that light textured soils like sands, there was no significant increase, which might mean that sandy soils have physical limitations for earthworms. This is reflected in the soil carbon as well, which in most studies it is intermediate, but you see it that if the soil C is very low, the effect is only marginal. We only had a small number of studies, 4, which have used soils with more C, which is not typical in cultivated soils, and you see it is variable then, but it is a small number of studies. What is nicer is rainfall had a nice number of reported studies, and you can see here that obviously where water is limiting reducing your tillage in some shape is very successful. Okay, pH was not that important. Plowing depth within the conventional made no big difference either, which basically means that if you have turning, action, it is detrimental to your worms. One that is of interest here is the length of the experiments, and you can see here there is at least a trend that, as you would expect, if it is less than 5 years, there is a smaller increase than when it is longer, but it seemed to indicate that this will actually level off, and then if it is longer than that, you might have maybe soil compaction and so on, which is negative. In here you can see we only found 2 reports which were longer than that, which were variable. One detail of estimating biodiversity, of course, is always methods, and there is enormous debates. Which ones are the good methods? Are they expensive? Are they fast? And so on. And you can see that most of the studies were formalin or hand sorting, or formalin and hand sorting, because most of the articles were sort of from the 1980s and 90s. There are some smaller ones which are used less, such as electrical, and now this is now a big debate. Is it because they are not efficient, or is it that formalin actually overestimates earthworm numbers in the reduced system? We also attempted then to split the types of tillage. Okay, so we had no-till, which includes direct seeding and so on, which has the biggest effect, and it seems that all the intermediate ones, which include minimum, reduced till, and so on, are less. You can also do this with biomass, of course, but here we would have less numbers of studies, and I will just skip this now in the interest of time. What you also have to do is, check if there is publication bias, which you would expect that, studies which were non-significant are less likely to get published, but we have no evidence of that, which is maybe because we also included PhD thesis and so on. If you do this work, you will see there is a lot of poor reporting, missing soil descriptions, and even the type of implement used and so on, which we will have to improve. So if you have more results, okay, that we have missed, or if you live here in particular, we would be interested in if you could send those. If you have articles published without errors, you are still in with a chance, okay, if you send them in. Okay, so that was that. I hope you've seen what you can do with this tool. Of course, it will never replace the expert opinion and the insights. And these are authors who have assisted us. Thank you very much.